It should start. Yes, it's there.
So I hope everybody had a really good lunch. We're extremely lucky to have Dr. Roland Langrock from Bielefeld University here to talk about hidden Markov models. Roland is one of the world's leading experts in this field. It's a highly varied field. Hidden Markov models are applied in a lot of different contexts, but more and more they're really important to animal movement. And I'm really happy to have Roland here. I've been really looking forward to hearing this talk. Roland, thank you so much for joining us, and really hope everybody enjoys hearing about hidden Markov models and animal movement data.
Yeah, thanks very much, Robert. Thanks everyone for coming. Nice to see so many people in real life after those last 2 years. It's really nice to be here. I hope you enjoy this. It's a bit statistical, but I'll try to make it as intuitive and easy to follow as possible. So just to quickly introduce me, I mean, there was an introduction, but I prepared my own introduction, so I would like to say I'm a professor of statistics and data analysis at Bielefeld University in Germany. So I'm a statistician working on method development, so I have expertise in particular on one class of models, which is a very specialized class of models, and that's the hidden Markov models. And those hidden Markov models, they turn out to be very, very useful in ecology, for analyzing movement data, also for capture-recapture data, occupancy data, all sorts of things. And that's basically the reason why I'm here, because I've been working in this field, statistical ecology, or just ecology, movement ecology, for about 10 years now, helping people to analyze the movement data. So what I'm going to do now the next hour and a half is I'm going to give you a very quick, superficial overview of what these models are, why they are useful models for animal movement data, and I will also showcase, using a very simple example, how you can fit these models in R. And then after those hour and a half, you might conclude this is not for me, I don't need that, that's fine, but maybe some of you actually feel that it's a good class of models to know about, and then you can go on and learn more about them. Okay, I've never given a course on HMMs this short, so I can't really cover that much, it will be very superficial. Okay, so in terms of the material, and you should use this information now, make a photo or just write it down. So all the slides are on this web page, and maybe you can just open that web page now and open the slides now, because on the slides in particular there will be some R code, like snippets of R code, very short R code snippets, which you can just copy paste to try things out yourself. So if you have the slide, that also means you have the code. There's also the direct link to the code. If you just want to use the code, it's down here. So it's not very much, but you can, I mean, it's always nice to play around with the code and see for yourself that these things actually work. Right? So that's the slides, some literature. If you want to go on and read more about HMMs, there's the book that I co-authored. If you're interested in that book, just drop me a line and I will send you the PDF, so you don't need to buy that book. I don't think I'm supposed to do that, but it's too expensive anyway. Then there's this paper by McClintock et al., recent paper, which is a very good starting point if you want to see in what kind of areas HMMs are applied within ecology, like capture-recapture, movement ecology, and so on. That's fine, actually. And then finally, there's one which is more focused on software, which is by Thierry Pillow, which is on the Move HMM package, which I'm going to use today. Okay. So motivation introduction, we start with a simple example, and this will be like the running example. We will talk about this example throughout this course, and I'm afraid it's not a fish. It doesn't look like a fish anyway. It's a muskox. This muskox was tracked on the east coast of Greenland, and we will look at this example track, but I mean, it could be any animal. It could be an elephant, could be a lion, could be a shark, could be some sort of fish, could be anything, right? It's more about the patterns that we see in the track and how we deal with those patterns, and it happens to be a muskox here that we're looking at. So this is the track that we're looking at. It's not a very long track. I wanted to have things very simple here. So we're not looking at multiple animals and several months of tracking. We just look at 2 months of tracking, so 60 days for this muskox, and it's hourly data. So we have 60 days of hourly XY positions of this muskox in Greenland. That's 1,440 positions in total. That's the length of this time series. Okay. Always takes a bit, I click it and I have to wait 2 seconds. So why should we actually bother trying to find a statistical model for muskox movement or any movement track, any animal? So what are the point? What are the possible objectives of such an analysis? So here's just a quick overview of the kinds of things that I usually aim at trying to learn about animal movement. So there are like general patterns that we might want to learn something about. How many behaviors does an animal exhibit, for example? That's not always clear, whether it's 2 or 3 or 4 behaviors. And then how much time is spent in the behaviors, how long do animals travel, how long do they rest, and so on. And then also how that depends on things like the environment. Anyway, and then the more interesting questions, that's the ones down here. This is about how animals respond to their environment mostly, but also how internal drivers, sex, age, fitness, and so on, how that affects the movement. So that means basically covariate effects of, I don't know, environmental covariates and movement. Okay. So this is the track again, now without the map, so that you can see it more clearly. This is the track, these 1,440 XY positions of this Muskox. And if we just look at this from a statistical point of view, obviously we're now interested in seeing or finding out or discussing what kind of patterns we see here. Right? And from a statistical perspective, what I see is that sometimes we see very long steps, so very long distances covered within an hour. So here, for example, the animal is moving along the coast, it's clearly going somewhere. So this is relatively fast traveling, and it's very directed traveling. So this would be what we call like a correlated random walk. It's strongly correlated in the directionality, and also there's relatively fast movement. And then at other times, like here, for example, or here, and very clearly here, of course, this is more like area restricted search. There's clearly some activity, some movement. So the animal is not sitting still at some point. There's some movement, there's change in the coordinates, but it's not directed in any way. So probably there's a nice foraging patch, and the animal moves around on that foraging patch. So we see clearly different behaviors, and that's the whole idea, the whole reason, the whole motivation for why we're using hidden Markov models. So hidden Markov models, what they're going to do is they're going to translate this idea that the movement is composed of different behavioral modes. They're going to translate this into a statistical model. So the model that we will see now in a second will exactly correspond to this idea. We have 2, 3, 4 different behavioral modes. And they somehow determine what we actually see in terms of the movement. Okay. So first thing we do is we will not model XY coordinates directly. So we will not have as the time series that we model the bivariate coordinates, the XY coordinates. We could be doing that, and there are models that do that, like stochastic differential equations. But when you think about it, when you look at your Excel file and you have the XY coordinates, you look at the coordinates, then you don't see what's going on. You don't see what the animal is doing. The coordinates directly, they're not really informative. What is informative is the steps between the coordinates. How fast, or how far did the animal travel? And how much directionality was there in the travel? So between coordinates, so over time, are there longer steps, and also are the turning angles, the changes in direction, are they large or are they small? So what we then do is we take the coordinates, so here's the coordinate, next hour, next hour, next hour, and as the first thing what we do is we convert this to a time series of step length. So step length is just the distance traveled in any given hour. So this would be the step length in the first hour, second hour, third hour, and so on. We will later extend this to also look at the directionality, not just how far they traveled, but also how much persistence there is in direction. So that will then be, okay, so here we have a relatively large turning angle, another relatively large turning angle. We will incorporate that later on, but just to get started with a simple model, we just look at steps. So distances traveled in this sequence of 1,440 hours. Okay, so this is how you load the data and prepare the data in R. So by now, I mean, if you're trying to follow this in R, you will have the code by now, I hope. So you download the data from a web page, and then there's this package which we are going to use here because it makes our life much easier. That's the move HMM package, which Theo Michelot mostly wrote with a little bit of help by myself, and also Toby Patterson. So that's the package we're using here. That's the most simple package there is for HMMs, but there are more general packages also. You need to install this in R, you load it, and then there's this function prep data, which for you makes this transformation from coordinates to step lengths and also turning angles. So you start with a matrix of coordinates, x, y coordinates, and what this prep data gives you then in the end is a series of step lengths and turning angles. As I said, we are not yet dealing with the turning angles, but we will later on. So now we're mostly interested in the series of step lengths. So we have the first hour, second hour, third hour, 2 kilometers traveled, 2 kilometers traveled, 2 kilometers traveled, and here it's unknown because somehow there's a missing observation here. No satellite reception, whatever, for the GPS. Okay, so there's some missing data points, but in principle, we're looking at this series of step lengths here. Okay, so you can look at the data yourself. And then this is how it looks like. This is now the time series of step lengths, the 1,440 observations that we made over time. Okay, so first hour, second hour, and so on. And here you see the step length in meters. So these 3 locations where the animal traveled 2 kilometers at the very beginning, and then sometimes it's only a few meters, and then sometimes up to about 3 kilometers. And again, now statisticians, as we are, some of us anyway, we look at this and we ask ourselves, what are the patterns here? What are the key patterns? What are the patterns that we would like to translate into a statistical model? Okay, statistical models is all about having a simplified, abstract representation of the data generated process in order to learn something about what's going on. So what are the patterns here? Well, the patterns are, as we saw from the track itself, that sometimes the animal is very active, moving a lot, and also in a very directed way, which we don't see from here, because with that we would need the turning angles. But just from the step length, we see that the animal is traveling relatively long distances here, 2 kilometers in a given hour. I don't know anything about musk oxen, but I guess given the size of the animal, 2 kilometers is actually quite remarkable for that animal in 1 hour. Okay, and then later in time, here for example, we have many hours, many days actually, where the animal in any given hour never traveled more than 200 meters. So maybe it found a nice foraging patch here and just stays there for 3 days. Okay, so it looks like clearly there are at least 2 distinct behavioral states, like a very active state where the animal travels from one foraging patch to the next. This is what we see here, and here, and here, and also there. And then another state which is more like area restricted search, maybe combined with the resting. And in fact, when you zoom in, when you look more closely at the time series, you see that here, actually, in this period of time, there must be 2 different behavioral states, because sometimes there are like 4 or 5 hours here, where there's basically no movement at all. This will just be GPS measurement error, most likely. And then at other times, like in this period, there is some movement. The animal is not sleeping. It's not sitting still somewhere, but it's also not a very long step. So here we have step length of up to 5 meters, and this is maybe 100 meters, 150 meters or so. So at the closer inspection, we see really 3 behavioral states, which I would interpret as a statistician, not a biologist, but it should be resting. This would be resting. This is something which we would probably call area restricted search. And then those things here we would call traveling. Okay, so that's the first idea. Well, and the second thing to observe here, is that these different behavioral modes, 3 of them, let's say, they occur in clusters. So when the animal is in any behavioral mode, it tends to remain in that mode for some time, usually like 5 hours, sometimes 10 hours. We saw that just now with the resting state. When the animal is resting, no movement at all, and we tend to see this for 3, 4, 5 hours. And likewise for the area restricted search or foraging, and likewise for the foraging, for the traveling. So these are the patterns. Different modes leading to different magnitudes in the step length, and those modes, they occur in clusters. That's the patterns that I see here. Okay, so this is just a summary of what I said. So we have certainly high and low activity, and at close inspection, low activity is actually 2 different modes, like no activity at all. That's the resting mode. Step length of up to 5 meters, and some moderate activity, which is maybe 100 meters, 150 meters of step length. Okay, so we see actually 3 levels of movement activity, and that we would like to translate into a model. And we would also like to translate into our model that these behavioral modes tend to persist over time, for some hours typically. Not necessarily always 5 hours, maybe sometimes 2 hours, maybe sometimes 10 hours. Okay, so those are the patterns. Different behavioral modes and clustered behavioral modes. And then we already have the first slide where I actually tell you what an HMM is. It's pretty much exactly what I just said, but focusing on the biology. This is now the pretty much immediate translation of what we said into a statistical model. We have different modes leading to different step length magnitudes, and those modes occur in cluster. And now, what is an HMM statistically? So what is the statistical model here? We have an observation process. This is the time series that we observe. So this is what we have here at the top. This is the observed time series. Here's the step length, the next step length, the next step length, and so on. So 2 kilometers, 2 kilometers, 2 kilometers, 100 meters, 5 meters, and so on. That's what we observe. And then we assume that there's some mathematical model, a data-generated process behind this, and that works as follows. Every of these observations, every step length, is basically generated by the underlying behavioral mode. So this is the behavioral mode underlying that step length, this is the behavioral mode underlying that step length, and so on. Okay, so the animal might be traveling. So then here we have larger step length generated. Then the animal switches to resting, and then we have lower step length generated. That's the whole idea. And these arrows just indicate dependence. This step length depends on this behavioral mode. This step length depends on that behavioral mode. And there's also dependence over time in these behavioral modes. So that when the animal is traveling here, most likely it will still be traveling here, and most likely still here, and then at some point it will switch the state. Okay, so this step length depends on the current underlying behavioral mode, and that mode actually depends on what was the previous behavioral mode, because they tend to persist over time. So that's the idea. So let's summarize that. Each of these observations, each step length, we assume to be generated by one out of n possible distributions, and n is the number of behavioral modes. So think of 3 here. For the muskox, n is 3. So that could be 3, right? And then we have three such distributions. Let me paint them here, maybe. So we now need a distribution for the step length. So here's the step length, and we now need 3 different distributions. So when the animal is traveling, we would say that the step length is typically maybe a kilometer, 2 kilometers, maybe 2.5 kilometers. So we will have some distribution which could look like this, which will then generate us a step length of about, I don't know, let's say this is 1 kilometer here. Okay, and then here we have 2 kilometers. So this could be the distribution that generates the step length whenever the animal is in the traveling mode. And then we have another distribution which corresponds to area restricted search, and that would generate step length which are much shorter, maybe like this. Step lengths in the order of 200, 300, 400 meters. Actually, it's a bit too wide to the right, it should be a bit more to the left, but just a sketch. And then we have a third distribution, and that would be the distribution which is active whenever the animal is resting. And that will be a distribution which is more looking most likely, oh, hang on, it's difficult to draw here, more like this, the blue distribution. It will only generate step lengths of about up to maybe 50 meters, okay? So we have different distributions depending on what behavioral mode is active, resting, foraging or traveling. Resting would be the blue distribution, foraging the orange, and traveling the red distribution. Okay, that's something we assume. And then depending on what behavioral mode the animal is in, either the blue distribution will generate the step length, or the orange, or the red. And this process down here selects which of these distributions is active at any point in time. So we could have, for example, we could be starting in a traveling mode. So we are here in state 3, yeah, the red state, and then state 3 again, and then state 3 again, which means that here we have the red distribution generating this step length. And then we see something like, I don't know, for example, 2 kilometers and 1.8 kilometers and 1.5 kilometers. And then the animal switches, and maybe afterwards, after the traveling, it's exhausted, and now it switches to the resting state, and it remains there for some time. Which then means that we have this blue distribution from the top right, generating us smaller step lengths. So that could then, for example, be just 5 meters or 7 meters, GPS measurement error, basically. That's the idea. So we have the sequence of behavioral states, somehow generated so that there's some persistence in the states. And then depending on the state, either of these 3 distributions will generate the outcome. That's the whole idea. Is that clear regarding the intuition? Okay. It usually takes some time for these models to really be understood because it's relatively complex in terms of the dependence structure, but I hope that the basic intuition is sort of okay to follow.
But
yes?
you pick the distribution of the model that finds those states. You tell it is going to be 3 states, find it? Or you...
I mean, we will talk about this, but we will estimate everything, right? We will estimate everything. We will estimate these distributions. It's just parameter estimation, how they exactly look like. We will estimate the dynamics here of how the states evolve over time. The one thing that we don't estimate is the number 3 here. This is something that we actually select in advance. We look at the data and make up our mind what we think. Is it 2 states or 3 states or 4 states? And then we fit the model. But of course, it's something which in the end is just model selection. You can fit a 2-state model and then a 3-state model and a 4-state model and use statistical criteria to select between those models. But once you have selected some number of states, let's say 3 for the muskox, everything is then estimated. This is just a sketch of how the model works, but how the distributions actually look like is estimated from the data. Okay, this is then how the fitted model actually looks like. We have not yet talked about estimation, but I wanted to give you some idea how the model then eventually looks like. So this is the model fitted to the muskox data. These are those 3 distributions. Apologies, the colors are not the same, but it's the same idea. So we have the 3 states, State 1 would be the resting state, so you have step length of up to maybe 20 meters only. So all the step length generated whenever the animal is in the orange state, in the resting state, they're all small. And then sometimes the animal is in the blue state, that would be the foraging state, and the step lengths are between 0 and 400 meters, if you look at the blue distribution. And the green distribution, that's the traveling state distribution. It's difficult to see here, it's difficult to scale this so that it's easy to recognize, but the mean step length here in green is actually 800 meters. It's not a great visualization, but the mean step length here is 800 meters. So that's the mean step length when the animal is in the traveling mode. That's one part of the model. That's the part concerning how these distributions actually look like, conditional on certain states. But we also need to somehow estimate the dynamics of the state process here. How do these states evolve over time? And what we do there is we simply estimate the state transition probabilities. So we estimate the probabilities of switching between the different states. So from any state, you can switch to any other state at any point in time. So we estimate the probability of switching from 1 to 1, from 2 to 1, from 2 to 3, and so on. That's 9 transition probabilities in total because we have 3 states, 3 times 3. And then we summarize this in the so-called transition probability matrix. If you know Markov chains, you will know this. If not, it's still, I think, fairly intuitive. We have this 3 by 3 array, where in the first row, we have all the probabilities of switching from state 1 to some other state. So this is probability of remaining in state 1. This is the probability of going from 1 to 2, and this from 1 to 3. Second row is the same when you start out in state 2. So switching from 2 to 1, remaining in state 2, and switching to state 3. And third row, likewise, for the third state. So we have 9 parameters here to estimate. Actually, it's only 6 because there are some constraints. It doesn't really matter. There are 9 values here that are of interest to us, and those are the estimates. And then you see here, the interesting thing here, and that's very, very typical feature or pattern in HMM, you have higher values on the diagonal, because the values on the diagonal, these are the probabilities of remaining in a state. So this is the probability of remaining in the resting state when you are currently resting. This is the probability of remaining in the foraging state when you're currently foraging. And that's the probability of remaining in the traveling state when you're currently traveling. So probabilities of about 80% of remaining in a state. And that, of course, then leads to clusters. If you simulate data from this, like if you just generate a random state sequence at any point in time, regardless of whether you're in state 1, state 2, state 3, at the next time point, most likely with 80% probability, you will be in the same state. And that leads to this clustering of states. Okay? And I think this really nicely translates these key patterns that we directly see when we look at time series of movement data into a statistical model. Different modes, different behavioral modes, and they occur in clusters. These are the different modes. Different modes lead to different distributions for the steps. And then this is what we need in order for the modes to occur in clusters. Okay, and from this we don't yet learn very much. Right? This is just a basic model to get started. But then, of course, we will bring in covariates and random effects and stuff to actually learn something about the movement dynamics. But that will be the end of the presentation. Okay, so for now, it's just a basic model. Okay, any questions so far regarding just the intuition? We will now talk about slightly more mathematical way of defining the model and about parameter estimation. But just the intuition, maybe? Questions on that? Yeah?
So the different states are always based on, or they can on just have affect one step. So what, for example, if there's a pattern that is always 2 different steps, so there's some behavior where, I can't give you an example now, but there's some behavior, for example, where they make a small step and then a big step, and they repeat this. Could this be translated in a state, or can't you do it because the state is always one step?
I'm not sure I understand the question, to be honest.
So you have the underlying states, right?
Yes.
That then generate the steps.
Yes.
What if you have a state that actually would generate a pattern of steps?
Well, the pattern of steps implies that you have multiple steps, right?
Yeah.
Multiple steps. I mean, so you could do that in principle. You could have, like, for example, you could have a state where you say this state is basically active for a complete hour, for 1 hour, but within this hour I actually have 10 observations. that would then mean that this observation is a 10-dimensional observation. Sorry, that was a very mathematical answer, but I think I know what you mean, and the answer to that is, yeah, sure, you can have like one state active for a certain period in time, and that generates a whole pattern of movement. That is possible, but it leads us to much more complex model formulations. So it's not really a good point in time to discuss these in detail. No, it doesn't mean it's a good question.
I was just wondering if this is a restriction of the modeling of the model, or if you can make it more complex.
Basically, there are hardly any restrictions. So the things that you observe here, right now, these are things like 2 kilometers, 1.8 kilometers, and so on. Actually, these could even be curves, right? You could have, depending on the state, you could have observed a whole curve. That would be functional data analysis. Right? This is something Eric would be interested in if you see that. Yeah, so anything you could have here. Given the state, you could observe anything. It could be a sequence of things, it could be one observation, it could be a vector of observations, it could be a curve, anything. Basically, pretty much anything. It's very, very, very flexible. This is the most basic model formulation, and we can extend it in all sorts of directions. Yes?
Short question, if you can go to the next slide, back to the next slide quickly. Are there general rules or patterns that you see in these state transition probabilities such that it's commonly, or it's uncommon to switch between state 1 and 3, but more common to switch between 1 and 2?
There is a relation to the temporal resolution of the data. So if you have a very high temporal resolution, which you often have with acceleration data, like whatever 20 hertz or so, then sometimes you have a specific structure where you never switch from state 1 to state 3 because there's sort of like a transition. So whatever, if you have acceleration data and state 1 is sort of resting, and state 2 is normal swimming of a shark stay, and state 3 is a predation event, you would never go from resting directly to predation. It would always be like sort of a transition, and then you would have the pattern in this particular example. If this is a shark resting, this is a shark normal swimming, this is a predation attempt, then this would be 0. You never switch from resting directly to predation. But that's basically due to the temporal resolution that the animal, not within an instance, can't switch within an instance from 0 to whatever 20 meters per hour. Yeah, but so there are patterns, but for hourly data or relatively coarse data, you would not really expect strong structures here, particularly strong structures. The key structures we always see is the high values on the diagonal.
Yeah.
And they actually, the higher the temporal resolution is, the higher these values are, of course. Because if the muskox tends to sleep for 5 hours, and now you go from hourly data to observations once per minute, so you have 60 observations per minute, then you would have 0.99, 0.99, 0.99 here.
So does that inform the resolution that is optimal to use for these, or is it agnostic to that in some way?
From the perspective of the statistical model, the resolution doesn't matter. The resolution really is only relevant in terms of what behaviors you can infer and what you can't infer. The coarser the resolution, the more difficult it becomes to really pinpoint behaviors exactly, of course, because then at some point things start to overlap. Within an hour, the animal can rest for 50 minutes and travel for 10 minutes, right? So the coarser the resolution, the more unclear the whole matching of these states to actual behaviors becomes. You know what I mean, right? So the higher the resolution, the more clear the patterns become that we tend to see. But the model really doesn't care about the resolution.
Yeah. So if you had position data once a minute, would you retain that or prefer to subsample it to once per hour?
That only depends on what you're trying to do with the data. If you want to infer predation attempts, then you need the highest possible resolution, almost. Like, I don't know. If I look at shark data, for example, we often have acceleration data, 20 hertz. 20 hertz is too much. We don't need 20 hertz. But maybe 1 hertz is actually a good idea, rather than going down to 1 observation per minute, because predation attempt only lasts for a second or two. We would not see it if we like average over 1 minute intervals. But these are things, I mean, if I work with somebody like you, biologist with real data, we would discuss these things at length in detail before we start any modeling, what resolution is actually appropriate given the research question of interest. Eric.
Yeah, so one thing I see the issue is that if your number of states are increasing, you're estimating an unconstrained matrix of a growing number of calls. Is it standard or is it possible to impose structure on transition matrix? So basically saying like you'll never go from resting to sprinting. Or
Not really in animal movement, because in animal movement we tend to have either 2 or 3 or at most 4 states, usually. I mean, you will find animals at high resolutions where you can identify 4 behaviors, but you have to really think about what kind of animals, what kind of data you need to identify more than 4 behaviors. So I would say in the literature, 80% of the HMM papers, they have 2 states. And the remaining 20% is pretty much only 3-state models. So, and in that case, it's not that many parameters. You don't need to make additional assumptions in order to reduce the number of parameters. In other fields, like finance and other fields, you could have 10 states, 15 states, and then you might want to think about restrictions or lasso kind of things, regularization, but we don't need that here. It's getting a bit advanced also now. Okay, let me move on a bit, because it's too slow anyway. So that looks technical, but it's actually not that technical. This is just a mathematical way of writing down what the state process is. What is the state process? It's a Markov chain. It's a Markov chain, which means that we have this sequence of states, which are such that the state at time point t is determined probabilistically by the state active at time point t minus 1. So if we know this state, say this is state 2, then we know the probabilities of being in state 1, 2, or 3 here. This is what we have in this transition probability matrix. In this transition probability matrix, let's move back to the concrete example because it's easier. We have the structure here. So if we know that we are in state 2, then there's an 80% chance that the next state will be 1, 80% chance that we will remain in state 2, and 2% chance that we switch to state 3. So in here we have these probabilities that tell us how the state sequence evolves over time. If we know now we are in state 1, then this tells us everything we need to know about what the next state will be, what the next state will be. Of course, probabilistically. If we know we are in state 1 now, we do not know deterministically the next state will be 2. We just have the probabilities of the different states. Okay, but we need those transition probabilities and we summarize them in this so-called transition probability matrix. So in the example before, this was 3 rows, 3 columns. In general, it's n rows and n columns because in general we can have any number n of states. We also need the initial state distribution. That's not very important. This is just the probabilities of starting in state 1 in the very first hour of the time series, starting in state 2, state 3, and so on. So we need to sort of initialize the process. We need the distribution of states at the very first time point. Without this, the model is simply not complete because we need to start somewhere. But this is not of interest, actually. This is of interest. This controls the state dynamics, how the states evolve over time. Okay, so we sort of had that already, but I wanted to now, before we just had the intuition, so this again is the intuition as a reminder, we have these n component distributions, 3 in the Muskox example, so we have 1 distribution for the step length when it's resting, 1 when it's foraging, and 1 when it's traveling. In general, it's n distributions, in the example it's 3 distributions. The state process simply selects from those distributions. At any point in time, we are in state 1, 2, or 3. Given we are in state 1, we have a certain distribution for the steps. State 2, we have another distribution. State 3, we have another distribution. And how these states evolve over time, that's determined by this Markov chain, by these transition probabilities. So we always need these distributions, which tell you how the step length looks like, conditional on the state, conditional on resting, conditional on foraging, conditional on traveling, and you need these transition probabilities. Those are the 2 components, right? One component, this component is that's these distributions, and these arrows here, these are the transition probabilities. There's another slide with a more mathematical definition, but I'm going to skip that here, because to understand now what an HMM is, you don't need the more mathematical definition. It's there for completeness if you want to know about it. Okay, so if at this point you think HMMs are great, I need this, I want to learn about this, so here's the bad news now. Most of you are fish people, I take it, and fish data is actually usually, or very often, difficult for us, because we need, for an HMM to be usefully applicable, we need regular data. Regular in the sense that we need to have some sort of regular sampling unit. 1 observation every minute, or 1 observation every hour, or 1 observation every day. You don't usually have that. You have your acoustic telemetry, the fish swim around, and then we have an observation after 3 minutes, and then maybe after 7 minutes, then after 20 seconds. That's not a regular sampling protocol. You get observations whenever you have a signal, basically. That's opportunistic data. You get them whenever they come close to the antenna, right? So that's not a regular sampling protocol, and we have problems with that. because those transition probabilities, probability of switching from state 1 to state 2, for example, they need to be interpreted relative to the sampling unit. Okay, so if I have a probability of 17% of switching from state 1 to state 2, that means that's the probability of switching from state 1 to state 2 over a 1 hour time period. But if that time period is variable, sometimes it's 2 minutes, sometimes it's 17 minutes, sometimes it's an hour, we don't have a meaningful interpretation of these transition probabilities. So we need regularly sampled data. So we like hourly data, once per minute, 20 hertz, whatever, as long as they're the same length of the time intervals between observations. That's nice. What also works sometimes for marine mammals, for whales, so they come up to the surface to breathe, they go down, they come up, they breathe, they go down, and so on. That's also sort of a regular pattern. Then we often have observations whenever they come to the surface, and then the sampling unit would be one dive. We have a dive-by-dive series. That's a meaningful, I would say, regular sampling unit. We have observations every time the animal comes to the surface. And then this whole thing, the probability of transitioning, for example, from state 1 to state 2, would be interpreted with respect to this dive-by-dive basis. Probability of switching from state 1 to state 2 from one dive to the next. But there's no such unit for acoustic telemetry data. Okay, so what we then need to do, or what you need to do in order to work with these models, and people do that all the time, it's not like it can't be done, but it's sort of like not so satisfactory. You need to first of all make irregular data out of your irregular data. So that means you need to somehow interpolate or infer or filter your data so that afterwards you have data on a regular temporal grid. That could be hourly data or data every 5 minutes or data every 2 minutes. And that's always a bit tedious, always a bit annoying, because it seems a bit arbitrary what kind of time resolution you then pick, right? If you have basically data coming in all the time, different time interval lengths, it seems kind of arbitrary to then decide, or like, let's filter the data so that afterwards I have one observation every minute. Why not every 2 minutes? Why not every 30 seconds? So there's sort of like an arbitrary element to that. But again, I would say, what kind of resolution is adequate? Well, it depends, A, on the research question, obviously, what kind of things you want to infer with this model, and B, at what kind of resolution you can meaningfully interpolate. So if you have basically observations coming in roughly every 30 minutes, sometimes 40 minutes, sometimes 20 minutes, sometimes 50 minutes, it would not make sense to interpolate so that afterwards you have one observation every minute. I mean, it's not going to work, right? If you have 2 observations per hour, it doesn't make sense to try to make 60 observations out of that. Right? This is basically magically creating data. So it should be somehow matching to roughly the average time interval length between observations. Okay, should I do that?
Yeah, please.
Okay, so that's the warning. But there are people who know how to do this. I mean, Henrik, I guess, is going to talk about that when he presents his YAPs. And YAPs can do that, I guess.
No, not really. It depends
Okay.
more on the transmitters if they have fixed birthdays or not.
Okay, so forget about YAPs then.
Yeah.
But there's still crawl. There's still crawl. For example, there's an R package that can do that. And sometimes even just linear interpolation will be fine. Okay, so just as a warning. Okay, parameter estimation. Let's talk about parameter estimation. This is sort of like, this is going to be the most technical part. And I thought about sort of like skipping it all together basically and just telling you there's an R package that does it for you. But that would be, I don't know, that would be, yeah, misleading. It would be misleading because the thing is, the models are fairly easy to understand. They are fairly intuitive. Most people, when you explain them for a while, they understand roughly what these models are all about, what the idea is, what you can do with them. But the parameter estimation is really tricky. It's not like a linear regression model where you have your LM function in R and everything works magically and it's fine. It is a bit more tricky than that. We have to maximize the likelihood, there's some numerical issues, and you need to have at least a little bit of appreciation of the fact that this is numerically difficult, because otherwise it's going to be very frustrating. Yes, Milan?
Just one question. Where do you in this process do these angles? So far it was
Not yet. Not yet. Eventually, so later on, like in half an hour from now, so far we had the states and the steps. So step 1, step 2, step 3, and later on we're just going to add the turning angles here. So that at every point in time we observe a step and a turning angle. So it will then be a bivariate time series or 2-dimensional time series, whatever you prefer. This will be the extension then to model steps and turns. And of course, we will want to model steps and turns because the turns are meaningful. The steps alone, we lose information. If we just consider the steps, we lose information on the directionality of the movement. So this is what we will want to do, but I want to have things simple first. So this is why I focus just on the steps first. And then we build up complexity. Okay? Okay, so how does it look like in practice? What do we actually do when we work together? Like Felicie and I, we discuss whatever Langrock data, we want to fit HMMs. What do we do? Well, we first of all look at the data, we look at histograms, we look at the time series, we look at the tracks, and from that, we do several things. Well, actually, mostly two things. Well, we have to select on these component distributions. So we have to select what kind of class of distributions could be appropriate for modeling the step length. Right? So that means we look at the histogram, for example, of the step length, and then we see it's continuous, the step length, it's non-negative. So normal distribution is probably not a good idea, because normal distribution can generate negative numbers, we don't like that. We need a distribution on the positive real numbers. So that would be the first step, and then we need to select some distribution of the positive real numbers. And that could be something like a log normal distribution, a gamma distribution, a variable distribution. Many of you will not have heard these distributions, or at least not know them in detail. Yeah, but that's okay. It's just some distribution that works for positive continuous data. And then the second thing we need to select, and we discussed this or we mentioned it earlier, we need to select on how many states we want to have. And if we work with whatever Felicie's Langrock's data, what we are probably going to do in the end is we're going to try out models with 2 states, with 3 states, and 4 states, and then afterwards use statistical criteria to select which of these models is best. Slightly more complicated than that in practice, but that's the basic idea, that you try out different models and then select the one that works best. Okay, and then so you have selected, say, gamma distributions for the step length, and we have selected that we want to have 3 states, because we think there are 3 behavioral states. Then we need to estimate those parameters. We need to estimate the parameters of the gamma distributions for each of the 3 states. We have 3 different gamma distributions, and we need to estimate the corresponding parameters. And we need to estimate the transition probability, so the probabilities of switching between those different states. Those are the parameters that we need to learn from the data. And that's the tricky part when we talk about that. There are packages for that, but it's still not completely straightforward. Model selection, no time for that. Model checking, no time for that. But of course, there are tools for that as well. I'm just not covering them here because I don't have the time. So we talk about parameter estimation now. Slightly tricky. We will do maximum likelihood estimation. We don't have to do that. We could also use MC-MC, which is fine. Many people use MC-MC. Maximum likelihood estimation tends to be faster, and that's the main reason why I prefer maximum likelihood estimation. So MC-MC samplers tend to be relatively slow for HMM. They're relatively slow to converge, and they're mixing issues. So they're mixing issues, and there's also label switching issues for those of you who are familiar with Bayesian inference techniques. All of this can be dealt with, but maximum likelihood estimation, at least for simple models, tends to be easier to implement and tends to be much faster also. So we focus on maximum likelihood estimation. If you've never heard of that, there's one slide here summarizing what maximum likelihood estimation is. The idea is that we try to find those values of the parameters, so the transition probabilities and the parameters of the gamma distribution, that make the data look as plausible as possible under the model. So we basically wiggle around our model until the model with that specific parameter values makes the data look as plausible as possible. Okay? So we know there are clusters in the data, so we will not have diagonal entries which are relatively small. We need higher diagonal entries in the TPM, for example. Okay? And then maximum likelihood estimation just simply means we look at the likelihood, which is simply the joint probability or the joint density of all the data points. So just the likelihood of seeing that very time series that we have, the time series of step lengths, and we regard this as a function of the parameters, and then we find those parameters for which we have the highest likelihood. That's maximum likelihood. Find those parameters which make the data look as plausible as possible. It's a very simple idea. It's simple enough that people actually find it just confusing. Why would that work? You just select the parameters that make the data look plausible. But it works really well. It's intuitively appealing. You want a model that makes the data look plausible. It's feasible. You just need to maximize this thing with respect to the parameter vector, the theta. Just some people will know that it's not always that easy, but in principle, usually it can be done, and it can be done with Markov models. And most importantly, it has very good desirable theoretical properties. The estimators are biased for large samples, unbiased for large sample sizes, consistent and things like that. Statistical properties which we simply like, but you don't have to worry about that. We don't know about these things. Okay, now this is the one most technical slide here, but I'm not going to go through the details. The bottom line here is, that there are really cool techniques, really nice techniques for calculating the likelihood of an HMM in a very fast way, which is not intuitively clear that this is even possible. Because so the likelihood is basically the probability of the observations. So the probability of your time series observations. Now, and in principle, in order to calculate this probability, you need to consider all the possible state sequences that might have led to the data. So you basically need to list all the possible state sequences, 1, 1, 1, 1, 1, 1, 1, 2, 1, 1, 1, 2, 2. And so you need to make a list of all the state sequences and calculate what is the possibility that this state sequence may have led to the data. And then sum over all those probabilities. This is actually what you need to do in order to calculate the likelihood. And that's a very, very computer-intensive task, of course, because if you have 1,000 observations, say 2 states, and when you list all the state sequences, that's 2 to the power of 1,000 possible state sequences. 2 to the power of 1,000. That's a very large number. No idea how large it is, but it's not something that you can easily calculate. But there are really magic tricks, really nice, fantastic magic tricks that allow you to still calculate the likelihood in a very fast way, in a fraction of a second, actually. The whole idea is, intuitively speaking, that you sort of like, you walk along the time series and you update the likelihood as you go. So you start at time point 1 and you calculate basically the likelihood contribution of that data point. Then you go one step further to time point 2, you update your likelihood. You go to time point 3, you update the likelihood. And that's what you see here in this likelihood formulation. This is the likelihood basically up to time point 1. Then there's an update, likelihood up to time point 2, and so on and so on. And that's the so-called recursive scheme. It's an iterative procedure where you update the likelihood step by step, going along the time series. And that's sort of magic. And that's the reason why HMMs are so popular, not just in ecology, but also in statistics in general. That you have a fairly complex model, which is also very flexible, and still you can very easily calculate the likelihood, and more importantly, you can very fast way calculate the likelihood. So even if you have, I don't know, actually I say here thousands, but I didn't revise the slide in the last few years. I could also write in the millions, right? Like the number of observations could be 10 million, and you could have 3 states, and you can calculate the likelihood maybe in 2 or 3 seconds, which is really nice. Okay? So I just wanted to have the slide so that you appreciate the beauty of this. Okay? Beautiful, yeah? Magic, yeah. So just make a note here, this is nice. We are happy about this, we're very happy about this. Not all complex statistical models are as nicely behaved as this when it comes to maximum likelihood estimation. So we need to maximize this thing, whatever it is really, I'm not talking about that. But happy to chat about this later. So we need to maximize this with respect to this parameter vector. And that includes everything. That includes the parameters of the gamma distribution, and it also includes the transition probabilities. So we need to find those transition probabilities and those parameters of the gamma distribution that maximize this expression. And that we have to do, unfortunately, and that's the difficult part, numerically. Numerically meaning that we have to somehow numerically, in some sort of approximate way, find the optimal solution to that. I give you a brief sketch of that because I'm pretty sure that not all of you will have heard of numerical maximization. So the idea is basically the following. If you have a likelihood function that looks like this, say, so this could be the likelihood function. So this could be the function giving you the probability of the data as a function of the parameter vector. That's what we do basically. We play around with our different theta values, the parameter values, and then we try to find that parameter value theta for which we have the highest likelihood. So the maximum likelihood estimate in this case would be here, because here we are at the highest point of the likelihood. This would be, not coming, showing up. I see it here, but I don't see it there. Ah, now it's coming. Yeah. So this would be the maximum likelihood estimate here, this value of theta, because here we have the highest value of the likelihood. That's the value of theta which makes the data look as plausible as possible. And that we need to find. We need to find that value, and that's the difficult part of HMMs. And numerical search algorithms basically the idea is the following. You start with a guessed parameter vector, so you guess where that maximum likely estimate could be, somewhere, right? And so we have a guess, and that could be, for example, say here. Theta naught, I call this. Theta naught, this is my initial value. And then what we do is, at this point, we look at the value of the likelihood function, and even more importantly, we look at the slope. We look at the slope, and then we see actually this is uphill if we go to the right, meaning that if we move to the right here, we're moving uphill, the likelihood is going to increase. Slope here is positive, so we move to the right. Okay, so we move to the right, and then maybe our next best guess would be this here, theta 1. So we go from our starting value theta naught, we update this, we come to a theta 1 based on just walking uphill. We walk uphill at the likelihood function. The likelihood function is basically a mountain, and we just walk uphill. So we are at theta 1, and then we see actually the slope is still positive. If we go further to the right, the likelihood will still increase. So we go a bit further to the right, and then maybe we make a step which is slightly too large, theta 2, and then we realize, oh, we went too far. Okay, so that was the peak of the hill, and I just went too far. And now the slope is negative. Okay, I need to go back. I need to go back, and I go the other direction. And eventually, I will then end up at this peak. It's like if I tell you find the highest peak here around Budweis, you just walk uphill all the time. That's the idea of numerical maximization. You just walk uphill all the time. That's all that you need to remember there. But there's one problem, and that's the key problem in practice, local maxima. And that's the problem the following problem. If your likelihood mountain, so this is my likelihood mountain, where we're finding the peak, but if our likelihood mountain looks like this, like a nasty mountain, many maxima, so if we start looking here then, theta naught, if this is our starting point, and we now basically walk uphill all the time, if from this point here, we always walk uphill, then we end up on this peak, which is not the highest possible peak. So that's a local maximum, rather than the global maximum, which would be here. So that's the kind of thing that we would like to avoid. How do we avoid it? We avoid it by trying to find good starting values. The starting values should be somewhere here in this example, in this sketch. In other words, if the starting values are too far off, if they're too far away from the good region, then we might end up in a bad place. And then this is like in practice one of the key problems that practitioners face with HMMs. The issue of selecting good starting values, because the packages, also the package that I'm using here, the package will do all of this thing here. All this here, the package will do for you, but you still have to select the theta naught. There's no package in the world that can do that for you, because it's just something. It depends too much on the data. It can't really be automized. That's the problem here. So we need always to think about this theta naught as a warning. Okay, just one slide on this distribution for the step length. So I mentioned the gamma distribution early on. Why? Because we need a distribution of the positive real numbers. Normal distribution won't work, t-distribution, all these things won't work. We need something which, I mean, we could use the gamma distribution, the log normal distribution, the variable distribution, the exponential distribution. There are many distributions, but all these distributions are not that well known, usually. But the gamma distribution is one that we like very much because it's sort of similar to the normal distribution, because we can parameterize the gamma distribution in terms of a mean and a standard deviation parameter, just like the normal distribution. There's a mean parameter. This would be then the average step length that we observe. Right? If the mean is 10, then the average step length would be 10. And we have a standard deviation parameter. But of course, it's not a normal distribution. As you can see here, in particular, it's not symmetric. Okay? It has all sorts of different shapes. It can even have the strictly monotonically decreasing density function, the black one, but very often for movement data we would see shapes like the orange, the blue, or the yellow. Okay, but it's usually it's right skewed. Usually it is right skewed. It's always right skewed, so it's not symmetric. Okay, and now we can fit our first model. So going back to the data, the Markov data, just to remind us of what we have there. So we now want to have a three-state model. Actually, let me check. No, I'm starting with a two-state model. I start with the simplest possible model. This is now about how we do this in R. So if I want to fit a two-state model, one gamma distribution for like an inactive state, covering all those observations here, and another gamma distribution for the larger observations here. How do we select this theta naught, the initial parameter value? Well, we need to select this mu for the resting state, for the inactive state, and also for the active state, for the traveling state. So we need two MUs, one such that we have all these shorter steps covered, and another one for these larger ones. So if we just look at the magnitudes here, these are all observations between 0 and 200, so let's pick something maybe 20 or 30 as the average step length in the inactive state. 20 to 30 maybe. And for the other state, it's a bit difficult without having the intuition, but I mean, these are the ones that you see immediately, just visually, those are the most prominent ones, but all these should also be in that state, all these more active ones. So actually, the average step length should be maybe about 500 or 600. Okay, so that would be two possible starting points for the MUs. So you start with the 20, 30 for the inactive state and maybe 500 for the other state. We also need to select the starting values for the standard deviations. I don't know, here, standard deviation could be 10 to 20 maybe, and in the other state we have a higher variance, so the standard deviation could be maybe 100 or 200. Okay, but this is all a bit hand-waving here. It's not like you need to select precise values. They just should be close enough, right? But it doesn't really matter in the end if you select 10 or 20 or 15. But what you shouldn't do is, you should not select a mean step length of whatever 5,000 as a starting value, because then you end up in the local maximum. So it should be close enough to what you expect is reasonable. So let's see what I did actually. So I selected as the starting values 5 and 200. So not quite what I just said, but I didn't remember what I did. Okay, so 5 is the starting value for the mean step length in state 1. Just the starting value. This is not a parameter estimate. This is just how I initialize my walking uphill algorithm. Okay, that's the starting value for the mean of the gamma distribution in state 1, and that's the starting value for the mean in state 2, the more active state, and 2 standard deviations. Here the rule would be usually to take 2 large values. So the larger these are, the wider these distributions are, and then that way you make sure that all the observations are covered, at least. Okay, so this is just choosing these initial parameter values. In principle, we also have to choose initial values for the transition probabilities, but that can be automized. And that can be automized because these transition probability matrices, they always have the same structure. They always have the large values on the diagonal. So this can be automized, and this package, the move HMM package, by default, it will do that for you. Okay, so you don't need to worry about that. You just need to select the starting values for the gamma distribution. And then we have this function fit HMM. You don't actually need this. I'm just doing that to measure how long the estimation takes. But fit HMM is the function in move HMM that will fit such a model to your data. So you pass on the data, you select how many states you want to have. So this is a number of states. This is the initial value. So here you plug in whatever you selected as the initial values. This doesn't matter. This is just about the output. And here I'm specifying that I don't want to have a turning angle distribution. Coming back to Milan's question, later on we want to have steps and turns. And by default, this function actually fits a step and turn model. The whole function, the whole package is designed to fit HMMs for steps and turns. But everything I've been doing so far is just steps. So for now, the distribution for the angles, there is no distribution. I'm not fitting any distribution to the angles. Okay? And then how does that look like? You get some output in the end, so you get the value of the log likelihood at the maximum, and you get the estimates. So you get estimates for the gamma distribution in state 1, so the mean of the gamma distribution in state 1 and the standard deviation in state 1. Very small values, so the mean step length in state 1 is actually just 4 meters. This is really the animal being inactive, just some GPS measurement error. And in the other state, it's 224. That's the mean step length in state 2, and the standard deviation. And we have the state transition probabilities, and we see on the diagonal we have 75, 85%. So tendency to remain in a state. And there's also this initial distribution, but that is not interesting. This is just the probability of starting in state 1 versus state 2 at the very first time point. Why would we be interested in that? But it needs to be estimated, because otherwise the model is not complete. Okay, so this is interesting, and this is interesting. Now local maxima, just to illustrate this, so what we then do in practice, so that you have heard it once at least, this will not be like the first thing you do yourself, but so you should know that this is what then happens actually in practice. What we do is we fit this model many times for lots of different starting values. So we just try out lots of different starting values. It's like when I tell you, Maya, whatever, well, you know the highest mountain here. Who's not from Czech? I don't know. You're not from Czech. Okay, so you need to find the highest mountain near to Budejovice. So what do we do, right? I make you start your search over there and over there and over there and over there. So you have 10 different starting points all over Budejovice, and from each of these starting points, you just walk uphill all the time. And then you have 10 different peaks. Actually, some of them might be the same peak, but sometimes you might arrive at different peaks. And from those 10 attempts of finding the highest peak in this area here, you will select one which is really the highest. That's what we do here. So I have a loop here, 20 times I maximize the likelihood, 20 times I randomly select initial values, run the estimation, and in the end I look at the corresponding likelihood values. So I have 20 maximized likelihood values here, but they're not all the same. Okay? So the highest one is this value here. This is actually what we saw here before. 7388, so that's the highest one. But there's another value, this one here, which keeps reappearing. That's the local maximum. So quite a few times, this algorithm actually gets trapped in this local maximum. Okay? But also many times it finds the global maximum. And this is meaningless. We're not at all interested in this. We're only interested in the global maximum. But important note here, the more complex the models become, if you bring in covariates and random effects and additional states, the more complex the models, the more of a problem this becomes. Because you then have a higher dimensional problem. Basically, this is just an optimization problem. Now I'm optimizing in 6 dimensions, because the parameter vector is 6-dimensional. If you bring in more stuff in your model, you have higher dimension. Higher dimension of your parameter vector, it's more of a problem. Yes?
Any reason these are negative numbers that I missed?
The reason is, so what is the likelihood? The likelihood is basically the probability of the data. It's not quite the density, but for simplicity, let's say it's a probability. So you have the probability of observing your 1,440 step length. Okay, what's the probability of 1,440 step lengths? Well, the first probability will be something smaller than 1, and then for the second step length, it's also smaller than 1. So you have to imagine, so each step length basically has a probability of smaller than 1 of being observed. And the likelihood is a whole product of this. So you multiply the 1,440 numbers with each other, which are all smaller than 1. So you get an extremely small value for the likelihood. It will be 0.00000, and after 100 decimal places, you will have the first value there. It's a tiny, tiny number. And what we actually maximize here is the log likelihood, because it's equivalent to maximize the log likelihood. That's equivalent to maximizing likelihood. And if you take the log of a tiny quantity, very, very minimally larger than 0, you get a value like this, minus something in the thousands. This could be minus millions. But the number itself is not meaningful, actually. The magnitude of this number doesn't tell you anything. This is just the logarithm of the probability of observing your data. But the more data you have, the smaller the probability will be of observing your data. If you have millions of data points, what's the probability of observing exactly those million data points? It's tiny. It's really tiny. And if you have billions of data points, it's even tinier. So the more data points you have, the larger in magnitude will be this negative number. But that's not meaningful. There's no information in this number in itself. It's only in relative terms. This number is higher than this number. So this is a better value of the likelihood than this. This is a global maximum, this is a local maximum. But the number itself, no meaning to that. It was a bit technical, but yeah. Good question though. Okay, so here's the fitted two-state HMM. We saw the three-state HMM before. This is the two-state HMM now fitted to the muskox. You have the resting state distribution, the orange one. So we have steps of up to maybe 15 meters. So this is really all the inactive behavior of the animal. And then the blue distribution captures everything else in this case. We saw earlier on that we actually want to have 3 states. So what happens when we fit a 2-state model is that the active state, the blue state, captures everything which is not inactivity. So both the area of restricted search and the traveling state, this is all summarized under the blue state, which is not ideal because actually there was a difference between the animal moving a little bit in some foraging patch and moving a lot along the coast when it's traveling. So we do want to differentiate these two behaviors. This model is not sufficient here. And then when we go to the 3-state model, you saw that graphic earlier on, but then now we have the orange state pretty much as before, the inactive state, but the other state, which was before, the blue state, now becomes two states, namely the blue state going up to 400 meters, roughly. This would be the area restricted search state, some activity, but not directly traveling. And the green state, hard to see, but the mean step length here is 800 meters. The information is not on the slide, but I know it. The mean step length in the green state is 800 meters. So this is really the traveling state. Just a second example, I wanted to have at least one fish in here, so there's some shark acceleration data here, and also to showcase that it's not always steps and turns, right? We can also look at acceleration data, for example. So this is acceleration data collected for an oceanic white-tip shark, and what we have here is the overall dynamic body acceleration observed over time, and the resolution is 3-second intervals. So this data has already been downsampled. I think the actual resolution was 25 Hertz, 25 observations per second, and it was downsampled by a factor of 75 to 3-second intervals. Simply because if you look at this, there's so much information in there, you don't need a higher resolution than that. You see what's going on. I don't actually know, Felicia, you're the shark person here. But for me, I don't know what all this means, but I believe that these kind of things is probably normal swimming. Then you have like bursts, whatever predation attempts. But I'm not quite so sure what that actually is. Maybe some sort of gliding? I don't know, we can talk about that later. I would actually like to know. But it's very clearly 3 different behavioral modes. If you look at the time series, also if you look at the histogram of these observations, the overall dynamic body accelerations, even from the histogram, you can kind of guess that there must be 3 behavioral modes. This here would be the behavioral mode corresponding to the probably normal swimming. And all here in the right hand of the distribution, you have all these bursts in activity. And this is here, those lower values, whatever that actually is. I don't know, I'm not a shark person. Well, I'm a shark person because I'm a shark. I don't know much about shark biology, I just like sharks. Okay? Anyway, so this is the data we want to analyze. This is the time series of acceleration data for these sharks. So how do we fit an HMM to that? Well, based on that, let's go through this step of formulating models. What would we do here? Well, I would like to have 3 states for this, clearly. I would like to have one state to capture all this normal swimming behavior. I would like to have one state to capture the bursts and one state to capture this, whatever that is. Okay, so 3 states. What distribution do we use? Well, before we had the gamma distribution. Now, what do we have here? We actually do have negative values here. That's because I took the logarithm of these overall dynamic body accelerations. Okay, so these are actually real numbers. I can use the normal distribution for that. So in this case, the distribution conditional on the state, so within the state, that distribution can be a normal distribution, which is actually a bit easier than the gamma distribution. You could also do this for the step length, actually. You could take the logarithm and then use normal distributions. Okay, this is the fitted model. These are the fitted 3 distributions, 3 normal distributions for the normal swimming, the blue one, the bursts, and the last one, the unclear one. And it fits the data almost perfectly. It's like, I mean, when you look at this, to me, this looks like cheating. Somebody must have simulated the data. Somebody must have simulated these data because it looks too good to be true. You don't usually see that good a fit of an HMM2 data, but in this case, it's just perfect. Okay, so that's the 3-state normal HMM, Gaussian HMM, because we have normal distributions in there. This is actually something that you could not do with the move HMM package. With the move HMM package, you can only do steps and turns, gammas and bigos and whatnot for the steps, and some other distribution for the turning angles. But of course, there are packages that allow you to fit such a model, like this model here. And there's Momento HMM, which is sort of building on Move HMM, but allows you to do many more things, including fitting Gaussian HMMs. There are more packages, even more general HMM packages, but these are not written for ecologists. They're not tailored to logical data. So they're just generalist packages. And of course, you can work with those, but they tend to be less intuitive because they're not designed to ecologists. This one, and also this one, these are really written for practitioners working in ecology. And of course, you can write your own code if you are very quantitative and you like to code. So people like myself and also my group, some of them are here, we would always write our own, not always, but pretty much always write our own code, because it's actually not that hard once you get the hang of it. How long do we actually have time? Do we have 90 minutes or 2 hours?
Well, actually, we should start a coffee break at 3, so 20 minutes. But I guess that we can, you know,
yeah, okay. It's fine. I think I spent, as expected, a bit more time than planned on the basic model, and I think that's most important, to get the idea, right? What the main idea is. Like, why is this model usually a good fit to movement data? And now I will probably speed up a bit so that you see all the rest, but I will not spend as much time explaining the details. So this is now like a quick tour of all the other things that you can do with HMMs. So state decoding. State decoding simply means you have your time series of steps, for example, and then you actually want to know what did the animal do at any point in time. Did it travel, did it forage, did it rest? So that's state decoding. You have your time series and you want to decode the underlying, the hidden states. And there are two ways you can do that. Slightly mathematical slide, but there will be visualizations in a second. You can globally decode, which means that you find the most likely sequence of states to have generated the data. And the output of this, if you do this, the output of that will be a state sequence like 1, 1, 1, 2, 2, 2, 1, 1, 3, 3, 3, 1, 1, 1. This is the output of that. If you do global decoding, that will give you the most likely state sequence to have generated the data. Okay? And the output is simply whatever that most likely state sequence is. Local decoding, there you look at individual time points. So maybe I'm interested in hour 27 for whatever reason. And for that hour, I then calculate the probability of being in state 1, the probability of being in state 2, the probability of being in state 3. We could spend some time discussing this. There's a subtle difference between the two. They're not exactly the same things. And it actually depends on what you're trying to do with this, whether you use global decoding or local decoding. I'll show you the output, then you know the main difference between the two. Okay, this is how it looks like in Move HMM. You fit your model first. So this is always conditional on a model. So you find the most likely state sequence, for example, given a model. You need to first of all fit the model. And then you do the decoding. And the algorithm for that is called Viterbi. That's simply the name that finds the most likely state sequence. That's the Viterbi algorithm. So you apply Viterbi to your fitted model object. Okay? And then you get the most likely state sequence. And state props, that would be the local decoding where you get the probabilities of the states. Okay, how this looks like. So this is for the Muskox data again. I'm zooming in a bit, otherwise it would not be easy to recognize anything. So I'm zooming in on the first 200 observations here, rather than 1,440. So you have like these 3 2-kilometer steps and then it continues, but only the first 200 hours. That was the fitted model, state 1, the inactive state, state 2, the active state. And this is now the Viterbi path, so the most likely state sequence, but I'm only showing the first 200 observations. And then you see obviously like these first observations here, they are allocated to the blue state, to the active state, and like here 50 to 70, this should all be inactive, so let's check. It's pretty much all inactive. There's some active, so that's this thing here. These few observations, which are maybe 100 meters, they were allocated to blue, but everything else was allocated to orange. So it does what it's supposed to do, right? I mean, no surprise here. But that's the output. That's fine. That's the output that you get out of this global decoding. Just the most likely state sequence. Local decoding looks similar, first of all, but the key difference is that here you actually get probabilities. Here you get at each point in time, you get the probability of being in state 1 versus state 2. So what I'm plotting here is the probability of being in state 2, the active state. And then you see at most occasions, in most hours where we have observations, the model is quite certain which behavioral mode was active. So whenever we are up here or down here, that means the model is certain that we are in state 2 up here or state 1 down here. But at some occasions, here, whatever, the model is not quite so sure whether the underlying state was state 1 or state 2. And that's, of course, always then the case when we have observations which are sort of in between. So when you have an observation of, whatever, let's say 20 here, or let's say 15, this could have been generated by either the orange or the blue distribution. And then the model just doesn't know for sure. Of course, there's uncertainty, right? If there's a 12-meter step length, you can't know what the behavior was in that hour. It's not possible. It could be either of the two. It could be a large GPS measurement error, or it could be the animal is traveling, but at the end of the hour, it just traveled 12 meters. Okay, so there's state uncertainty then. And that's the difference. Here you have the probability of the states. Here you just have which states are most likely. That's the key difference between the two. Okay, now multivariate time series, and this will now go towards this turning angle incorporation into the model. Actually, on the first slide, I have an even more complicated HMM here, but we will then talk about the steps and turns model. Okay, so often we have multivariate time series. So the typical example will then, of course, be steps and turns. So you have horizontal movement data, you convert them into the step length between locations and the turning angle, so the changes in direction between successive movement directions. Okay, that's what we usually do. Or you could have a whole bunch of statistics. So for example, here, this is a model that we fitted. Stina fitted that model, she's actually here, PhD student of mine. This is a model that we fitted to gray seal data in the Baltic Sea, and there we have a dive-by-dive basis of data. So we have basically one data point for each dive, but the data point is multidimensional because it includes the maximum depth during the dive time, the surface time after the dive, the horizontal step length performed while diving, and the turning angle relative to the previous and the next dive. So it's a 5-dimensional data point for each dive. So we have the first dive of the seal, 5 data points, 5 summary statistics on that dive. Then we have the next dive, again, 5 summary statistics on that dive. The more information, the better, right? We want to infer the behavior of the animal. Why would we focus only on the maximum depth, right? The horizontal movement also matters. The dive time, of course, will be correlated with the maximum depth, but not perfectly correlated. There might also be some information in there. Okay, and then the idea is that you simply add those additional variables to your HMM. And that's completely straightforward. There's no mathematical challenge in here. It's completely straightforward. If you understand the basic model, going from the basic model to this model, same thing. The likelihood looks exactly the same. The maximization is exactly the same. The only thing that changes is, of course, that you now need to think about 5 different distributions. You need a distribution for this, for this, for this, for this, for that. So what would then happen is this could be a gamma distribution, this could be a gamma distribution, this could be a normal distribution, and this could be some other distribution. You could have different distributions because they need to be tailored to whatever type of data you have. Right? If they're all positive and continuous, they can all be gamma distributions, but they might not all be positive and continuous. Okay, so let's focus on our classic example of steps and turns. So this is the matchbox again. Now here is the time series that you know very well by now. This is the time series of step length, and now I also look at the turning angles. Difficult to visualize turning angles, so you don't really see that much here. But actually, if you do look at, for example, this time period, these whatever 10, 15 hours where the animal was traveling along the coast, down here the turning angles, they're all very small. These are all close to 0. So pi is turning around 180 degrees, minus pi is turning around 180 degrees in the other direction. Okay, so whenever you have a large turning angle, here or here, that means that relative to the previous hour, the animal went in the other direction. And the 0 turning angle means it continued in the same direction. So from here now, next hour I'm here, and the next hour I'm here, that was a 0 turning angle. But if I turned around, that was a turning angle of either pi or minus pi, which is actually the same because turning angles live on the circle. So if I turn around minus 180 degrees or 180 degrees, it's the same thing. Mathematically, it's the same thing anyway. Okay, so 0 turning angles are interesting because 0 turning angles mean that there's directionality, strong directionality, persistence in direction, and larger turning angles, that's indicative of the animal not going anywhere. There's no directed movement, it's just, I don't know, walking around on grass, foraging patch, and feeding properly. Okay, so how do we model that? Okay, or how do we fit this model? I don't even have the model here, but the model is clear basically. I had it earlier. We have one distribution for the step length, gamma for example, and another distribution for the turning angle. Now the turning angle, as I just said, turning angle, that is something that lives on the circle. You need to have a distribution that generates values between minus pi and pi. And such that actually minus pi and pi is the same thing. So you need to have something which lives on the circle. And there are distributions for that, and the main distribution, the most well-known distribution is the von Mises distribution, which is actually a relatively simple distribution to understand. Because, so the idea is if this is the circle, and this is 0, and this is pi, so 0 means we're going straight ahead, and pi means we're turning around. So von Mises distribution is basically a normal distribution, but wrapped around the circle. So you have, if you have directionality, persistence in direction, you tend to walk straight ahead, then you would have a normal distribution here basically. So you would have like this Gaussian, the bell-shaped peak at the 0, so that most observations are close to 0, but then you wrap it around the circle like this. Oh, hang on, that was not very nice. Like that. So that most values generated from this distribution would be somewhere here, but you could also have some values there. Take the normal distribution, take the two ends, the left tail, the right tail, wrap it around the circle, make a knot, and then you have your nice wrapped normal distribution, which is then called the von Mises distribution. Also, we need to estimate parameters. In this case, we need to estimate also the mean, and the mean is some value between minus pi and pi. And then we have something which is called the, what is it actually called? The precision parameter, and the precision parameter is 1 over the variance. So this thing here, the precision parameter, I'm writing a standard deviation, but it's actually not correct. This is what confused me just now, sorry. This is the precision. The higher this value, the higher these values, the narrower this distribution is. So if you increase these values, then this von Mises distribution becomes narrower and narrower and narrower around your mean value. But again, you need to choose the initial values, and then you fit your model, and now I don't have this angle dist equals none, because now I am fitting in a distribution for the turning angles, and so then I have my steps and turns model. This is the output then, so what has changed? We have the TPM as before, the transition probability matrix. It's again 75%, 85%, not much has changed here. These are the two step length distributions, 5 meters in state 1 on average, 230 meters in state 2, pretty much as before, but now we have in addition the turning angle distributions. In state 1, we have a mean turning angle of roughly 3, which means that on average we are mostly turning around. And in the other state, we have something which is very close to 0 as the mean, which means persistence in direction. You move straight ahead. So how does that model look like? These are the two step length distributions, pretty much as before. They have changed slightly. And these are the associated turning angle distributions. So the blue is the active state, traveling and area restricted search. Then you see mostly turning angles close to 0, because here you have the highest density. And the orange distribution, that's the turning angles in the inactive state. Here you actually see mostly the animal turning around, most of the time. I mean, you still see also sometimes small turning angles, but the higher chances that you see turnings for 5 minutes. Okay, that will be the three-state model. I will skip that now. Well, one thing I want to say here. So the green distribution is the traveling distribution, and here you have an even narrower distribution now of the turning angles. If you look at this turning angle distribution, state 2 in the two-state model was capturing everything which is not inactivity, foraging and traveling. But because it's both foraging and traveling, traveling means you have 0 turning angles, but foraging means you could have anything, like a uniform distribution on the circle. And this is exactly what we see in the blue distribution. We have mostly zeros, but we also have some minus pi's and some pi's. If we do include a third state, then we end up with a clear traveling state, and then we have a turning angle distribution where you actually never see turning angles, which are corresponding to turning around, the animal turning around. You see really small turning angles now. So this is the really directed movement now. So clear difference between 2 and 3 states. And in this case, the 3-state model is actually much easier to interpret than the 2-state model. Okay, so this is where it becomes interesting, and we have 5 minutes for that. But that will do to give you an overview, covariates and random effects. All of this was just, first of all, finding a mathematical model, which I would argue in many cases is probably a good approximation of the true data generating process, the true mechanisms that drive animal movement. Obviously, some animal movements, certainly then also in 3-dimensional space and so on, they will be more complicated than what we saw for the muskox. But for the muskox, I would argue that the model that we just saw seems to be a good representation of what I believe would be the true underlying mechanism. Okay, but we have not yet learned very much about the behavior or interactions with the environment and so on. So this is where the fun now begins. Now we bring in covariates. And if you're interested in individual variation, you bring in random effects and things like that. So covariates, what do we do? Well, we can have covariates either in this process, in the step length process, or we can have covariates in the state process, in the behavioral modes. So we can have covariates like temperature, for example, habitat quality, things like that, whether it's raining or not. We can have these covariates affecting the step length distribution, or we can have them affecting the transition probabilities. We usually prefer the latter. We usually prefer having covariates in the state process so that the probability of switching, say, from resting to traveling depends on the temperature. So maybe that if it's getting hotter, then I'm more likely to switch to the resting state. This is usually what we prefer, both for conceptual reasons. It usually is more plausible from the logical perspective, But there are also statistical reasons which I don't want to discuss here because that would take me 10 minutes and I only have 3 minutes. Okay, so we want to have them in the state transition probabilities. And what we then do here, basically, you don't really need to look at the slide in much detail. We basically have within the GLM, within the HMM, we have a logistic regression. Because if I have a two-state model, this is what I have, a two-state model, a 2x2 transition probability matrix. If I am in state 1 right now, in the next hour, I could be either in state 1 or in state 2. So I have a binary choice. Do I remain in state 1 or do I switch to state 2? Binary choice. So it's a logistic regression scenario. We have a binary response, and the response here is whether you remain in state 1 or you switch to state 2. So that means we simply incorporate into our HMM a logistic regression model. That's all that we do, both for the switches out of state 1 and for the switches out of state 2. And that's what you see here. So we model, for example, this off-diagonal entry, the probability of switching from state 1 to state 2. We model this as the inverse logit applied to some linear predictor. This here, if you just look at that, this is exactly the formulation of a logistic regression model. Right? You have a linear predictor, and then you have a transformation, so that in the end you end up with a probability. And in this case, the probability is simply the probability of switching from one state to another. And then in this linear predictor, you can have anything. Temperature, time of day, you can have temperature with a quadratic effect, you can have random effects in there, you can have interaction terms, whatever you like. Yes?
Can it handle days with covariance? Or?
Very good question. No. It can't. If I do have NAs in my time series, I do need to impute some covariate values somehow. Because the thing is, in order to calculate the likelihood, I need to be able to calculate all these transition probabilities. Right? The likelihood, in my likelihood calculation, I need these matrices. And if I say my matrix depends on the temperature, but I don't have the temperature in some hour, I just can't calculate the matrix.
You can do a post hoc to calculate the probabilities and then superimpose the covariate after you have a probability and see if there's a correlation in that.
I didn't get that. Sorry. We can tell later. But so the idea would be that we do some imputation of the covariates. But if you have, of course, 90% covariates missing, then that's not a good idea to do imputation. So there shouldn't be too much missing covariates. Okay, so 60 seconds left. That will be fine for 10 slides. How this works in Move HMM, not much has changed. I just add this argument here, formula, and this is the formula where I specify how my covariate, in this case temperature, affects my state transition probabilities. So by default, what this will do is it will incorporate a logistic regression model into the state transition probabilities. And here you write the linear predictor just like you would do it in LM or GLM or LME4 or whatever you're using to fit regression models. So this is simple regression modeling syntax here, but within the HMM. And then you get some output, you get an intercept and a slope, of course, for these logistic regressions, and we can plot them. And this is how it looks like for the mass curves for the temperature. And what we see here, so this is probability of 1 to 1, 1 to 2, 2 to 1, 2 to 2. So the same arrangement that we have in the transition probability matrix, sort of like a 2x2 matrix. But now, of course, these probabilities depend on the temperature. And then what we see here is the probability of going from 1 to 2 increases when it's hot, but the probability of going from 2 to 1 also increases when it's hot, which is not a typical result, but that's what it is for this data. So what that means is this animal seems to switch more often between states when it's hot. Or in other words, when it's cold, it remains longer in whatever state it is in. It's longer inactive and it's longer active. And it's a small data set. I would not actually try to, I would not write a paper based on this. It's a data that's much too short on one individual, and I just want to showcase how this looks like in the end. You can also look then, you can do time of day, of course. You can incorporate time of day as a covariate effect. Then you need to just make sure that time of day, of course, you have periodicity. So after 24 hours, the effect should be where it started at 0.0, right? So 0. Midnight is the same as what it was 24 hours ago, so you should have the same. So you need to do this using something like either the trigonometric functions or cyclical splines, whatever you want, same that you would do also in the regression model. So you incorporate, for example, trigonometric functions, and then you get an output, that's the code, and then you get an output like that, where you have time of day now as a covariate, and it's estimated in a way so that after 24 hours, the regression function is exactly where it started. That's because of the use of the trigonometric functions. And from this, you can then also derive state occupancy, or the probability of being in states as a function of time of day. That's for the two-state model. So this basically tells you, during the day, the animal is more likely to be active. At nighttime, the animal is more likely to be inactive. Surprise. I learned a lot here. Three-state model is a bit more interesting, because here you see that in the early morning, like 6 a.m., 7 a.m., the animal tends to have, I mean, at least throughout the day, the highest likelihood of the animal traveling is in the early morning. Right? State 3, that's the traveling state. So the highest probability of being in the traveling state is early morning hours. The highest probability of being in the foraging state, state 2, is in around lunchtime and the afternoon. And then at night time, the highest likelihood, we have the highest likelihood of being in the resting state. I guess that makes perfect sense biologically, but I don't actually know, but to me it seems plausible that they would move in the early morning hours and then later on feed. Random effects, heterogeneity, individual variation, yes, we can do that, and I would like, I don't know, I can talk about that if you want, but I'm a bit running behind and I'm sure you want a coffee.
What do you want, guys?
Random effects.
If it's okay for you, Henrik, to start.
I think it's only one slide anyway, actually, I'm not sure. Check. Oh, it's 2 slides. Okay, random effects. So if you're interested in individual variation, so if you're either interested in individual variation, that is the focus of the study, or if you know there's individual variation and you need to somehow address it, you don't want to neglect it, well then you're talking about how can I now build my model. Ideally, you would still like to have a joint model for all the individuals, which somehow, however, incorporates this variation, which somehow acknowledges that there are differences across individuals. So in general, same like with mixed models, regression models, you have the different strategies. You can have complete pooling, where you say that all individuals are the same. Fit one regression model to all, one HMM or one regression model is the same thing, really, same idea for complete pooling. You fit one model to all individuals, and you don't allow for any variation between individuals. This would be fitting a linear model or a GLM without random effects, basically. You can have no pooling, that's the opposite extreme, where you say all animals are completely different. I don't see how I could fit a joint model, so I fit 5 different models to 5 different elephants. So you would fit 5 different HMMs, for example. So these are the 2 extremes. Either they're all the same, very simple to do that, obviously, you should just have the same model as before, or they're all different, which is also very simple to do, because you just fit multiple models. But neither of them is satisfactory, because in this case, well, you just ignore the variation, and in this case, you have the problem that in the end of the day, you have lots of models, and they might be very difficult to make sense of, actually. So imagine you have 5 different musk oxen, you fit 5 different HMMs, and those states, so what the resting state is for the first musk ox, could actually be different than what the resting state is for the second musk ox, and the third, and the fourth, and so on. Because those gamma distributions, they would now vary across the individuals. And that's not ideal. It's really difficult to make sense of these no pooling models where you have one model for each observation. And of course, also statistically, there's a disadvantage here, because you have then relatively few data points to inform your many models. If you have 100 individuals, you fit 100 models, that means you have just one time series to inform each model, so you have not as many observations to inform your parameters. So usually we want to do partial pooling, where we do have a joint model, so we just fit one model to the individuals, but we acknowledge that there's some difference between individuals, and that usually we do with random effects. Or sometimes we might also have individual-specific covariates, like the age of individuals, or the size of individuals, or the sex of individuals, and that might also explain some of the variation. So you could have, rather than the temperature that we had before as a covariate, you could have the size of individuals, or let's say the age of individuals. And the age could, to some extent, explain the variation across individuals, of course. If you don't have such covariates, then you need to look at random effects. So there's a summary of that. I think I've said all of that. So partial pooling is what we usually want to do. Just like with regression modeling, we also usually prefer partial pooling for statistical reasons and also for conceptual reasons. And there are different ways we can do that. We could say that, all the gamma distributions are the same across individuals. So what the resting state distribution, what the foraging state distribution, what the traveling state distribution is for different individuals, that's the same. but we could have the state transition probabilities varying across individuals, effectively meaning that we estimate one transition probability matrix for each animal. We can do that, but still then we have relatively many parameters because we need to estimate one such matrix for each individual. We could have, I will skip to this first because this is my preferred solution. If you do have covariates, which are individual-specific covariates like age, fitness, size, whatever, and those covariates you actually assume to explain most of the variation, then that's the easiest and best way to go, because that's easy to implement, that's easy to do, and also it actually tells you something about where the variation comes from. So that's really nice. If you don't have such covariates, or if they don't explain all the individual variation, then of course you're stuck with, mathematically it's tricky, then you need random effects. So you need to have basically a random variable telling you or governing the variation across individuals, just like you would do it in regression models. So you could have, for example, the gamma 1, 2 transition probability, so the probability of switching from state 2 to state, from state 1 to state 2, that could be a random variable where you have one realization for each individual. And we can do that with HMMs, but there's a major downside. It's very difficult to fit. Okay, and people like Kalina, who's sitting in the back, she's going to write a PhD on it. This is how difficult that is. It really is difficult. Final remarks. There's lots of stuff that I didn't cover, obviously, 90 minutes. We sometimes give this course over 5 days. So there's a lot more to talk about. I have a list here. There are lots of other ecological applications. Capture-recapture is a big thing, actually, for HMMs. There are many HMM papers on capture-recapture where you have, as the state, typically the animal is alive or dead. And the alive state could be further split in alive and healthy or alive and with disease. So you could have a capture-recapture model where you have the 3 states, alive and healthy, alive and disease, or dead. So that could be an HMM. Occupancy modeling is basically also HMM modeling, so you can regard this as an HMM. Yeah, all sorts of different applications actually. Forgot the other ones just now. Anyway, I did not talk about how you write your own code, how you implement the likelihood. This is what I would do in a longer workshop like this. Non-standard dependent structures, you can modify the dependent structure in all sorts of ways, bringing covariates in different parts of the model, have complex dependent structures where the observation in the current hour feeds back to the state in the next hour and things like that. Many things you can do, continuous time HMMs, Sina wrote a PhD about that, continuous valued state processes, so where you don't have 2 or 3 states, but where you have infinitely many states. So the state goes up and down, but continuously, gradually over time. Not so relevant in ecology, although there are some applications. Hierarchical HMMs, Timo wrote his PhD about that. This is when you have multi-scale data, so you have maybe one data stream which is hourly data, hourly GPS data, but then you have more high resolution data as well, acceleration data, for example, and you want to combine both of them in a joint model. So there you would have a hierarchical structure where you have one hierarchy level for the hourly GPS data and another level for the 1 Hertz acceleration data. Capital HMMs, I will skip those because they're not as important. And cautionary note, I think I've said this before, but the main downside of HMMs in practice really, for practitioners certainly, is that we have these numerical problems, which is really mostly local maxima. It really takes some time to get used to that. So when we give these workshops, and we teach people how to write this code, for example, from scratch, dealing with local maxima and selecting starting values, this is what people struggle with the most. But the good news is, if you do this for some time, if you fit a few HMMs to different data sets, eventually you really get the hang of it. And eventually you do this completely automatically in your sleep. But when you start with these things, this is the tricky bit. And most people, most practitioners certainly then do need help by people more familiar with HMMs. And if you work with HMMs and you do get stuck and you want help, then contact people like myself, like my PhD students. And there are many people now who do HMMs. 10 years ago, there were like, you would find 3 people in ecology working in HMMs. Now you would find 50, maybe 100 people. So it's getting more popular. Okay, so that's it. And yeah, drop me a line, come chat with me. Thanks for your attention and the questions. I'm sorry for shortening your breaks.
breathtaking display of HMs. I guess that we found ourselves in a need for HM, yeah. Everyone was. So I guess that we've got a short period of time
I'm happy
for
to
a
take it.
question. Okay, if not from this audience, maybe.
Henrik.
I'm sorry.
I want to know when we can join one of your week-long courses. That's allowed.
I don't know actually. So I should say that. I mean, so my whole group, so Sina and Kalina and myself, we like to give these workshops, and we do this for free actually. So I mean, it's not like we do this 3 times per year, but we like to do this maybe once per year. So if there's sort of like a crowd, a community, wherever, in Denmark, Norway, in Israel, wherever, if there's an audience of say 20 people who are really keen to learn about that, and we find the time, we are happy to come actually. We don't need to be paid or anything. If you cover the travel expenses, that would be nice, but we like to do that. Not too often, because there's always not enough time for things, but because the thing is, when we give these workshops, we usually initiate lots of collaborations over these workshops, so it's a win-win thing. I mean, people learn about HMMs, and we get to know people with cool data, and we initiate applied projects. So if you'd like to bring us to Denmark at some point.
I was here trying to find it out.
Yeah. Just yesterday, I said to Ran that I would like to give the workshop in Jerusalem because I've never been to Israel. So next year will be Jerusalem. Any other questions?
Yeah, are there things with regard to that as well? Are there like certain things that you see appearing every time when you get like fish data sets? Or I don't know if you have already a lot of examples of those where you worked on, but I guess there are some probably things or challenges that are always the same, like you say, intervals, but there must be much more others. And if so, then a workshop on this week.
Yeah. It's very good that you raised that. And actually, I should have thought about that in advance and have a slide dedicated to fish data, because fish data is different. It's different. It's not like Massbox. It's not as easy. No. Okay, fish data. So the first thing really is this irregularity that we typically have with your data. That's an issue which is a bit annoying for everybody. It's annoying for you to sort of like have to transform your data so that the models become applicable. It feels kind of weird, right? You would like ideally to have models that fit directly to your data rather than manipulating your data. And there are such models, but they are more complicated. That would be stochastic differential equations, and when you then want to bring in states, that becomes really difficult. Okay, so that's the first thing, this need to regularize data in time. And then other things are, of course, that fish also move in the vertical dimension in the water column. So it would usually probably not be sufficient to just look at steps and turns. You would probably also need to look at the vertical dimension, which in terms of the modeling doesn't create any problems. We would then simply use 3 variables, usually, steps and turns, to describe the horizontal aspect, and just vertical distance basically between current hour and last hour as the 3rd dimension. So the vertical distance covered, modeled with the normal distribution, for example, we could do that. And so that's another thing. So the model is slightly changed compared to terrestrial animals. And the last thing is, whenever I have worked with fish data, which is not very often, the patterns are not as clear as for animals like muskox or lions or elephants, and in particular birds. I mean, birds are the easiest ones for us, because they either fly or they don't fly. But fish are more difficult. For fish, my limited experience with fish data is that even if you look at the data, it's not so clear how many behaviors there are, whether it's even discrete, so whether behavior is even discrete, whether it's just resting and swimming, or whether it's more like gradual changes in activity levels. I don't know. So most fish data that I saw, the patterns are not as clear, which makes the models a bit more difficult to work with. But actually, maybe not the models, I mean the data is more complex. That's simply what it means.
I'm wondering if this might be due to the fact that we suffer from the fact that every position has probably, in some cases or a lot of the time, another error. So I guess I don't know about terrestrial telemetry, but if it's the case that for terrestrial telemetry, errors on positionings because of GPS errors are the same for all dots, then I can imagine even having the errors, you will see the states from the data. But I think for us, the states might be like covered or hidden below the errors maybe because, and also now, because technology allows us to have very short time intervals when we observe the fish, every error and every step might easily like be greater than the small time step. So the smaller the time step, the more of an impact an error becomes on the data. So, and I also wanted to ask, like, is there anything that can be done about this, like incorporating, if you would know the errors on each position, could you then incorporate this info and I don't know.
Dealing with measurement error in HMMs is basically not possible. And you then move on to state-space models. But state-space models, the models that are being applied in ecology, they don't have the behavioral state switching. It's just state-space models where they have the same structure as HMMs actually. You have an observation process, which is the actual position plus measurement error, and the state process is the actual position. It looks like an HMM, but actually you don't have the behavioral state. The underlying state is simply the true position, and the observation is the observed position, which is the true position plus error. So I mean, you can fit state-space models, but the objectives are different. They're completely different. It's just about trying to remove that error. It's like filtering the track so that you get a more realistic track afterwards. You can have state-space models with additional behavioral state switching, but then again, it becomes very complicated. People don't really do that actually, because it's so difficult. You need then MC methods, and they're terribly slow to converge. It's just a mess. Like 10 years ago, it was a bit of a thing in the community. Everybody was talking about using state-space models with additional behavioral state switching, but it kind of disappeared from the literature because it never really worked. So measurement error is something we can't really address. I would need you to somehow filter your data so that you're afterwards confident that the measurement error is negligible. And if it is not negligible, then I'm afraid I can't help with these tools.
Do you always have to work with a continuous data set, or could you have 1 day there, 1 day there?
Oh, that's fine.
That's fine?
That's fine. That's no problem whatsoever. So I mean for one individual though.
Yeah, because I was thinking of my data set because I wanted to use HTML when I was analyzing it. But then the problem was that we have to use the random things within the timing cloud, which made it, well, then I was like, okay, I tried a different approach. It was like for the master thesis, it was,
okay.
I gave up.
Yeah, but you can have multiple tracks, also multiple tracks per individual. So if you have one individual observed for 3 days and then you don't observe it for half a year and then you observe it again, that's fine. That would be fun. You have multiple time series then, but it would be pretty much the same likelihood. The product of the likelihoods of the individual tracks then, but that doesn't complicate things at all. I saw the chat popping up now and again. So are there any questions? I don't know.
We're invited to South Africa.
I am going to South Africa actually. Sina and I, we are giving a workshop in South Africa next month. Yeah, so we are coming to South Africa in Cape Town. Yeah, so sorry, I forgot about that actually. Somebody asked for a workshop. We're giving a workshop next month in Cape Town, but it's part of a conference, so I don't know. I think it's something you need to register for. But if you ask, I mean, I'm sure you can also join online virtually. I don't know. It's a 1-day workshop. It's not a 5-day workshop. It's 1 day. So it's a bit more extensive than this, but not much more.
And it's also free.
It's the ISIC conference. Sorry.
Yeah. Do we have some questions from virtual participants? Just raise your hand or place it in the chat, please.
Sorry, somebody said that they didn't hear the questions. Yeah, sorry about that.
That was before lunch. So it's a chat from the morning.
I have only one comment. Okay, fine.
Yeah, brilliant presentation.
Okay, should we then have people go get coffee?
Yes, I think so. Roland, thank you very much. Yeah.
Can I make a first question?
Sure.
Because I raised the hand up from the internet, but seems you didn't receive the. Okay. My question is about visual census surveys. I mean, sometimes happen that for some species, the only way that you can study them or harvest data more in general is through a visual census, in which you may record for single individuals, like time of diving, angles of turning, or what else, specific behaviors. Those kind of data are useful for these kind of models, can be used.
I'm afraid that I missed the first part, really. Sorry, I didn't understand the question, I'm afraid.
The fact is that for some species, you can't go into the field, or I have data set in which the survey for some species has been made by visual census, in which only like time of diving and turning angles and things like that have been recorded. Do you think they are suitable for this kind of models?
I don't understand the problem. I understood most of the words, but sorry, I don't understand the
Yes, yes, okay, don't worry. I will write to you.
Yeah, write me an email.
This way we can chat about it. In general, since it is evident that you can't hear well me, it's also the same for me. In fact, since this morning, I'm saying that here we have like acoustic problems, which is lack of,
Okay, just drop me.
Yeah, and the thing that we were asking is that if in general you can speak more near to the microphone. This way I can understand better.
Yeah, okay, Francesca, we will try to repeat the questions. Yeah, okay, thanks.
Okay, sorry, just send me an email.
Yeah, yes, thank you.
Okay, so guys, see you in 30 minutes. So enjoy the coffee. Yeah, starting, let's say, some 10 to 4.
Just before you all, if you want to follow along with the code while I'm running it, I will put the link to the GitHub on the screen. So you might come to the results guiding we will run the code.
But it was really, really bad.